Lokeshwari from 2nd MSc Microbiology, Serkatat College, Tirupattur. Here we are going to see about the sulfur. Sulfur is one of the most abundant elements on the earth, which is yellow, brittle, tasteless, odorless and non-metal. It is present in all kinds of the protein, and usually plants directly absorb sulfur-containing amino acids such as methionine, cytosine and cytosine. Sulfur is released into the atmosphere by the volcanic activities, burning of fossil fuels and decomposition of organisms. On land, sulfur is stored in the underground rocks and minerals. It is released by precipitation, weathering of rocks and geothermal vents. It also describes the movement of sulfur through the atmosphere, mineral forms and through the living things. Microbial transformation of sulfur are even more complex than those of nitrogen because of large number of oxidation states of sulfur, several transformations of sulfur and also occur spontaneously. Photosynthetic and chemosynthetic microorganisms contribute to the environmental sulfur cycle. The major steps involved in the sulfur cycle are sulfur dissimilation to hydrogen sulfide, oxidation of hydrogen sulfide to elemental sulfur, oxidation of elemental sulfur to sulfate, reduction of sulfate to hydrogen sulfide. First, sulfur dissimilation to hydrogen sulfide. Protein degradation in dead materials releases the amino acid which contains sulfur. Sulfur released from the amino acids forms sulfate, which is then converted to most reduced form, and the hydrogen sulfide is processed under a process called dissimilation, which refers to degradation of inorganic substance. Microorganisms involved in the sulfur dissimilation process are D-sulfur vibrio species, D-sulfur-bacter species and Archaeoglobus species in the anaerobic condition. Oxidation of hydrogen sulfide to elemental sulfur. Hydrogen sulfide is oxidized to elemental sulfur under various conditions. Oxidation of hydrogen sulfide to elemental sulfur was carried out under both the aerobic and anaerobic conditions. Aerobic condition, the main bacteria used are Thiobacillus species, Begotito species, Theothric species and aerobic anoxygenic phototrophs. And in the anaerobic condition, photolithotrophic sulfur oxidizers such as Chromatium species and Chlorobium species are used. Oxidation of elemental sulfur to sulfate. Oxidation of elemental sulfur to sulfate was carried out by anaerobic condition in the bacteria Allotromonas species, Clostridium species, D-sulfur vibrio species and D-sulfotomacallum species. This process involves anaerobic process in two steps. They are elemental sulfur is oxidized to sulfite, sulfite is oxidized to sulfate. Finally, return of phosphorus back to ecosystem through decomposition. Sulfates are reduced to hydrogen sulfide anaerobically by the means of D-sulfur vibrio, D-sulfuricans, D-sulfomonas species and D-sulfomacallum species. This reduction process majorly occurs in two steps. They are: firstly, sulfates are reduced to organic sulfur utilizing ATP by a process called assimilation, which are then referred to as formation of inorganic substance. Second step, reduction of organic sulfur to hydrogen sulfide by a process called mineralization, which means decomposition of organic substance. Finally, this is the complete process of sulfur cycle, and thank you so much for listening.